Good morning and welcome to hematology. This is our first course or our first unit on hematology, and we're going to focus a little bit about the MVP of hematology, which is our red blood cell. Today we're going to discuss the structure and metabolism of our red blood cells, the who's, how's, and what's of red blood cells, where they come from, and how they function. I'm sure all of you have learned about cell membranes in all of your biology classes previously. But today let's talk a little bit about the red blood cell membrane. The red blood cell membrane is a complex formation of glycolipids, glycoproteins, proteins, phospholipids, and cholesterol. And it's the combination of all of these components that enables the red blood cell to perform their function appropriately. So let's talk about each one of these things. So let's talk about the importance of the membrane complex and the way in which it combines. If you think about the environment in which if red blood cells reside, there are a lot of things happening. We have electrolyte balances changing. We have very narrow passageways through those capillaries. And so our membrane complex for red blood cells combine in such a way that they are able to be highly elastic and yet very stable. A healthy red blood cell membrane can respond rapidly to applied stresses of fluid forces. So if you think about changes in electrolytes where we may have a hypertonic or an isotonic or a hypotonic environment, depending on the salts and the other electrolytes that are present, our red blood cells need to be able to adapt to those stresses so they can swell or they can shrink accordingly. So a couple of other things about the red blood cell membrane. We know that it's semi-permeable due to that ability to allow fluid force across the membrane. We also have a lipid bilayer, and this supports the lipid bilayer, and there's supportive mesh-like skeleton that supports that lipid bilayer, and that helps maintain cellular shape, cell stability, and cell deformability. And those three characteristics of red blood cells are imperative for them to be able to perform their job. Cell shape is imperative because it allows a biconcave disc shape, which allows for a larger surface area to carry and transport oxygen and waste to and from cells. Cellular stability, obviously we want those cells to be strong enough to go through the circulatory system without rupturing or breaking. And then cell deformability is another factor that's very, very important for our red blood cells to be able to enter the smaller vascular structures within the body, such as those tiny, tiny capillaries. Let's talk for a moment about red blood cell lipid membranes. So there is a bilayer of phospholipids, and these phospholipids are oriented in a very precise pattern, which is critical to normal red blood cell survival in circulation. It gives it strength, structure, and helps maintain those fluid forces. It is interspersed cholesterol and glycolipids. So if you look at the diagram here, we've got some proteins at the top. We haven't really talked about them yet, but we'll get to that. And then we have this lipid bilayer. We've got the outside of the cell at the bottom, and they have those phospholipid heads and then a long lipid tail. And then we're going to have the inside where we have these interspersed bands of protein. Now, if this arrangement is altered, it could activate the clotting cascade and result in extravascular hemolysis. We will talk more about activation of the cascade when we get to our coagulation, but what you need to know is that phospholipids are, if they are not presented to the body in such a way that is recognizable, that it could trigger an abnormal clotting reaction. In addition to that, the glycolipids interact with other membrane glycoproteins to actually form many of the red blood cell antigens that are living on the surface of the cells. These are inherited through your parentage, and each person has a little bit of a different variation. You'll learn more about this in blood bank, but when we talk about blood types, not only are we talking about our ABO blood type, we're talking about RH, and there are multiple other antigens that are very specific to each and every person's red blood cells. And so those glycolipids that interact with the glycoproteins form those antigens, and that's why blood typing is more than just ABO blood type, we also have to sometimes be very antigen specific if any antibodies are formed against those red blood cell antigens. Red blood cell membrane cholesterol is in continual plasma exchange, so it's going in and out of the cell, into the plasma, into the cell, out of the cell, into the plasma, and so it's therefore affected by changes in the body's lipid transport system. So again, we've got a lot of different biochemical things that are happening there, and this is all due to the function of the red blood cell membrane. In some abnormal membrane lipids, we might see atypical morphologies. And if you look to the right, I have a picture here. These are called target cells. And these target cells are a result from the increased red blood cell membrane cholesterol to phospholipid layer. So the reason why we have an abnormal shape, we don't have the normal biconcave shape, but rather a formation of a target morphology. And again, this is due to an excess of cholesterol in that membrane. Now, I always think of these as they kind of look like donuts. So the way I remember the cause of target morphology is that donuts are fatty, full of cholesterol, and so are target cells. In addition to the lipids and the phospholipids within our red cellular membranes, there are a couple of really important membrane proteins. These membranes are asymmetrically organized in the red blood cell membrane. And the two most important, we have one called glycophorin. It's an integral membrane protein that makes up most of the membrane. And then the last is sialic acid, which gives the red blood cell its net negative charge. Now, if you think about magnets, a negative and a positive attract, but two of the same that are on the same pole, like two negatives or two positives, they repel each other, and that's really what we're looking for. This repulsive force is generated by that net negative charge on the red blood cell surface that occur due to the presence of the carboxyl group in the sialic acids, and that helps propel and push the red cells along through circulation, in addition to the pumping and the musculature of the vessels. Now, if we did not have those negative net charges, we did not have sialic acid as a part of our membrane, we would actually have a problem with clotting. And so thus, when these red blood cells have negative net charges, they repel each other along through the circulation, which is really helpful. We definitely wouldn't want our red blood cells to attract one another, or we'd have lots of problems with clotting. Another peripheral protein that's very important within the red blood cell membrane is spectrin, and this is important because it binds with other peripheral proteins to form somewhat of a skeletal network on the inner surface of the membrane to help the red blood cells maintain their biconcave shape, and also allows them to be deformable. Now, when I say deformable, what I want you to think about is a really thin frisbee. You know those cloth frisbees that are foldable? Well, this is what we need our red blood cells to be able to do. You can fold them, you can bend them, you can twist them. And if you look here, the spectrin is interspersed as this kind of long line. So if you think about it, it's like a rubber band. You can pull it, you can push it together, it'll retract, it will twist, and it makes the red blood cell able to get into the nooks and crannies of circulation where even the single cells at the end of our capillaries have to be able to receive oxygen and dispose of the waste, the cellular waste. And the spectrin within our membranes allows our red cells to be able to perform that duty. Okay, so one of the things that you might notice in my lectures is I really like to reiterate things. I like to introduce them, and then I like to go into more detail with them. So we're going to get into more detail about the red blood cell properties. We've already discussed deformability and permeability, but now I would like to go into a little bit more detail about each one of those characteristics of our red cells. When introducing new ideas or new concepts, I really like to give a visual example. So why deformability? Well, if you look into the pictures here on the right side of the slide, we can see blood flow as it goes from a larger vessel to a smaller vessel. This first picture here shows a blood flow of highly deformable healthy red blood cells and what it looked like, so they can kind of go into flatten down into this little tiny vessel here. And then this bottom picture here, we see cells that are hardened and less deformable. This could be an inherited problem. It could be a problem just because maybe there's an aging cell and so it's losing its deformability with age, but they become kind of hardened and they're unable to squeeze down into those little tiny vessels. And again, deformability is crucial to red blood cell survival through the microvascular, which is really important for oxygen delivery and cellular waste removal. So, why do cells lose deformability? If cells have proper membrane proteins, they have the proper ratio of phospholipids and cholesterol, they have everything they need to do. What happens? Well, if you hadn't noticed already, and we haven't really talked about red blood cell development yet, we're just talking about some of the characteristics. You will note that red blood cells are anuclear. They do not have a nucleus. They start out with a nucleus, but as they mature, they lose the nucleus and they have to utilize anaerobic respiration. So, one of the things that causes cells to lose deformability is the loss of ATP. We're going to talk more about how ATP is produced and those metabolic pathways later in this lecture. But when there is a decrease of ATP, that leads to decreased phosphorylation of the spectrin. Now remember, spectrin is the protein that helps the membrane maintain that biconcave shape and that deformability. We also know that calcium pumps within the cell membrane are dependent on ATP for function. They are not passive. And so as those calcium pumps can't keep up without ATP, calcium builds up in the membrane, it causes it to become rigid. And that decreases the deformability and the ability for our red cells to adapt with the shape. Healthy red blood cells, however, have enough ATP to survive the trip throughout the body while delivering oxygen to tissues and other cells and also removing cellular waste. As the cell ages and becomes less efficient in terms of ATP production, that's when we start to see the accumulation of that calcium within the membrane. So this might seem like a random slide, but I really am just showing you this slide to kind of show you some of the things that can cause a shortened lifespan. So our red blood cells typically have a normal lifespan of around 120 days within circulation. There are some things that can cause a shortened red blood cell lifespan, and some of them are inherited, like this condition known as hereditary spherocytosis, in which there's a lack of spectrin. There might be some abnormal inclusions, and what we have to remember is there are a lot of checks and balances within the body. Our splenic cords actually help pull damaged or abnormal red blood cells out of circulation. And that is because the splenic cords are kind of like in a network which makes passage impossible if the red cells lack that deformability. If so, those splenic macrophages will pluck them out and break them down for recycling. Unfortunately, when people have a large number of abnormal red cells, they're broken down far too quickly, and this can lead to anemia and other sorts of conditions that we will get into later on in our topic. Okay, so we have discussed in depth our deformability, but let's talk a little bit more about permeability. Our red blood cells must be freely permeable to water and anions, meaning chloride and bicarbonate. This helps maintain the pH of the blood so that humans can survive. Our blood pH for survival is a very, very narrow window. When you talk with Jenny in chemistry and you go through your blood gases unit, you're going to talk all about it. But our ability to exchange oxygen, water, carbon dioxide, bicarbonate, all of those things keep our blood pH in balance. And it also enables our red blood cells to function appropriately. A red blood cell must also be impermeable to cations and maintain its volume half-time exchange, so greater than 30 hours. And when I say cations, I'm talking specifically about sodium and potassium. There are very, very specific ratios of sodium and potassium intracellularly and extracellularly, and we're going to talk about those in just a minute. Okay, so ratios to remember. So we have an intracellular to extracellular ratio for sodium. We have internally, so inside the cell or intracellularly, we have 1 sodium for every 12 sodium molecules outside of the cell. So we've got sodium knocking on the door, but we have very selective cation pumps that only allow a certain ratio or certain number of guests to enter. And then as that guest enters and leaves, another one will be entered in. So it's really important to maintain that balance. This is part of our electrolyte balance for healthy cells. Our intracellular to extracellular ratio for potassium is actually the opposite. We have 25 guests at the house party inside the cell of potassium, and then one lonely guy outside knocking to get in. And that is why hemolysis is such a problem when doing analysis within the lab, because when those red blood cells rupture, all of that potassium that is intracellular now goes out into the circulating plasma. And unfortunately, it can artificially elevate the potassium in terms of our analysis. So really, really important that we have good samples simply because we don't want our potassium and other electrolytes to be out of balance when we do analysis. And again, these are facilitative. They have to go through cation pumps within the cell membrane. Now that we've talked about the importance of the red blood cell membrane and the importance that it plays in the function of the red cell, we need to really look at the core of the function. We know that red blood cells carry oxygen to other cells in the body and that they remove waste. But how do they carry oxygen? Well, every red blood cell has what we call hemoglobin, and you may remember this from your biochemistry class. A healthy adult hemoglobin molecule contains 2 alpha globin and 2 beta globin chains. That's the protein portion. And then we have a heme. We have 4 heme groups that are also included, and that's going to be made with a protoporphyrin ring and iron. And this is where the oxygen actually binds. So oxygen is attracted to the iron, it binds to the iron and is able to be circulated in that way, and then thus distributed to other cells in the body. Unfortunately, there are a few conditions that can cause an abnormal synthesis of hemoglobin. So let's talk kind of about what normal hemoglobin production needs in order to be successful. Very first thing is we need adequate iron delivery and supply. So we need a warehouse of iron and we need transportation for that iron. We need adequate protoporphyrin synthesis and we need adequate globin synthesis. So if any one of those things is disrupted or abnormal, we cannot get a normal hemoglobin synthesis. Right, so let's talk first about iron supply and delivery. We have a very specific transport protein for iron called transferrin, and transferrin delivers iron to the red blood cell membrane. In mitochondria, iron is inserted in the porphyrin ring to form heme. Now wait a minute. You might be asking yourself, if red blood cells are acellular or anuclear, how are their mitochondria? Well, During the production and development of red blood cells, they are nucleated, and at that period of time when the iron or when the hemoglobin is being developed and inserted into the red cell, it is still nucleated, and so therefore there are mitochondria present at the time. Excess iron aggregates are kind of stored in kind of readily available iron source called ferritin. So transferrin is a transport protein, and ferritin are the aggregates of ready to use iron supply. And about 2/3 of our body supply of iron is bound to heme and hemoglobin. If you think about that, that's pretty amazing. All of the iron that's in our body, only 1/3 of it is in storage, and the rest is in red blood cells being utilized for oxygen transport. And we will go into more depth about our carrier proteins later on and talk about exactly how they work and where they're at. So just to give you a brief overview about protoporphyrins, why do we need them? Well, we need protoporphyrins in our body to form the heme groups that are within the hemoglobin molecule. And this, of course, occurs in the mitochondria, like we discussed. We have glycine plus succinyl CoA, and that equals something we call DALA or DALA in the mitochondria. In the presence of the correct enzymatic processes, protoporphyrin precursors are formed. So we have porphobilinogens, and there's a condition that can occur called porphyrias. You're going to learn all about porphyrias in chemistry, but when you get to your enzymatic disorders, but we are going to just kind of briefly talk about it because it is something that can affect our protoporphyrin ring production. So porphyrias can occur if some of the normal enzymatic steps are blocked. And then we see things like darkening of the skin and the eyes and discoloration of the body. But again, you'll learn more about those mechanisms and porphyrias when you get to your chemistry rotation. Okay, so we've talked about our heme groups, and now we're going to talk about our globin chains. So if you recall, you're back to the days of your biochemistry class in college, you're going to remember you guys probably talked a little bit about hemoglobin and porphyrin chains. So globin synthesis is really important because it occurs on red blood cell specific cytoplasmic ribosomes, and these arise from structural genes that we inherit. Each gene makes a very specific polypeptide, and each somatic cell, including red blood cells, has these genes. So we typically have 4 alpha, 2 zeta, 2 beta, 2 delta, 2 epsilon, and 4 gamma. Now, you might be asking yourself, well, I thought we only had alpha and beta chains. Well, yes, as an adult, that is a normal hemoglobin or a globin chain for adults, but there are a variety of different combinations of these globin chains throughout development from embryonic through adulthood, and we're going to talk about the types and percentages of hemoglobin presence at various stages of development in the next slide. And you will want to pay close attention because these are often the things that are going to be on exams or maybe even board exams. So really know your fetal development, your types of early hemoglobin, and kind of how those transition from embryonic stages into adulthood. And it's really important to know those because as we get into more detailed hematological diseases, we might have to understand how the globin chain abnormalities cause those to occur. Okay, so let's talk first about these early types of hemoglobin. So we have 3 different types of embryonic hemoglobin, and these are going to be dependent upon what phase of embryonic development a person is in. So we have the first type, which is called Gower-1, and that is going to consist of 2 zeta and 2 epsilon chains. So instead of the alpha and beta chains, we have zeta and epsilon. Next, Gower 2, which is 2 alpha and 2 epsilon. So now you've seen that transition from zeta to alpha. They both have 2 epsilon chains, but we're kind of moving forward. And then we also have Portland, which is 2 zeta and 2 gamma. So it's kind of interesting there. Instead of alpha, it's back to zeta. At about 6 months of fetal development, maybe a little slightly earlier, probably 5 months, between 4 and 5 months, we have a full-on development of fetal hemoglobin, and this is going to be called hemoglobin F. And this is where we start to see the first part of our hemoglobin very consistently made with 2 alpha globin chains. So in fetal development, around between 4 and 5 months, we have 2 alpha and 2 gamma chains. And this is pretty persistent. And actually, once the baby is born, you'll start to see a mixture of hemoglobin A, which is our adult hemoglobin, and fetal hemoglobin. It'll kind of fade away. And once the baby's between 6 months to a year old, you won't really see that hemoglobin F presence anymore. But you will see it early on in the infant stage of development. Okay, types of adult hemoglobin. So there are a couple of different things. Now, I just told you that hemoglobin F is not persistent past the neonatal stage, once a baby becomes 6 months to a year old. However, there might be a tiny bit of residual hemoglobin F, and in some conditions we're going to see higher percentages of hemoglobin F than are normal. And so I put this into the slide so that you would see that it is possible to have some hemoglobin F even as an adult, but the percentage is very, very minuscule unless you have a hemoglobinopathy or something that's causing abnormal hemoglobin production. The majority of normal hemoglobin in our body is hemoglobin A, and again, that's going to be our 2 alpha and our 2 beta globin chains. So that's going to make up a normal hemoglobin A. We have a subtype of hemoglobin called hemoglobin A2. So again, we have 2 alpha and 2 delta. And then hemoglobin F we already talked about was 2 alpha and 2 gamma. Now I want you to take note here. Once we have exited embryonic and fetal development, we now have alpha chains in every type of hemoglobin. So alpha chains are imperative to normal hemoglobin development. And we do have a couple of things that can affect the production of alpha chains, which can be imperative or detrimental, should I say, because those alpha chains are imperative to make any type of hemoglobin at this stage of development. So let's look to see how our heme and our globins are kind of combined to make a functional molecule. So each synthesized globin chain links with the heme to form hemoglobin. For example, you're going to have 2 alpha and 2 beta chains plus 4 heme groups. Now remember, our heme group is going to be made up of our protoporphyrin ring plus our iron molecule. That's where our oxygen is going to bind. The precise order of amino acids is actually crucial to the ability for the hemoglobin molecule to survive. So we might have a substitution in one of our hemoglobin molecules or one of our globin chains, and we are actually going to end up with something that could cause, for example, sickle cell anemia, and that is based on one tiny amino acid substitution within that globin chain. We're going to talk more about our hemoglobinopathies later on, but just know that any one substitution, it's going to change the shape and the function of that protein. Okay, so just a brief overview. Our normal hemoglobin function with our 2 alpha and our 2 beta chains are going to be delivery and release of oxygen to tissue. So not only does our hemoglobin function as a delivery system, but it also has to understand when to release the oxygen from that molecule. And again, the facilitation of CO2 excretion is imperative to help maintain our pH and to rid our body of cellular waste. All right, so we've talked about the binding of oxygen to hemoglobin, but let's talk about how the hemoglobin or the red cell knows when to offload oxygen in time of need. And that is due to the credit of a little molecule called 2,3-biphosphoglycerate, or sometimes called 2,3-BPG. You may hear it called 2,3-diphosphoglycerate, which those two nomenclatures are really interchangeable, but it's important to know that they are the same thing. You may hear it used one way or the other on board exams or in policies and procedures. So the importance of 2,3-BPG is its function in offloading oxygen. Okay, so oxygen is unloaded to tissues when spaces between the two beta chains widen. So if you look at this picture, we've got so picture to the left, we have oxygen with our O2 bound, and this is just our oxyhemoglobin. But what if our body needs oxygen to be released? Maybe we're running a race, maybe we have an increase in metabolism due to the heat, maybe our pH is off, and we really need to have more oxygen released. We have this little molecule 2,3-DPG, which comes and it wedges itself between our two beta chains, which causes the conformational change to squeeze together, which is called a tight binding structure, and pop off the oxygen molecule that is bound at that point. So beta chains are really important to be able to allow 2,3-BPG to kind of wedge its way in there. We're going to talk more about metabolic influence to produce 2,3-BPG, but we're just introducing it now, so bear with me. When hemoglobin loads O2 and becomes oxyhemoglobin, the beta chains pull together, so it opens its mouth, kind of looks like a little Pac-Man. And when there's an increased need for oxygen in the body, 2,3-BPG will come, wedge itself in there, and pop off that O2 molecule so that it can be distributed to cells that are in higher need. Now we're going to get into more detail about that here in just a bit, but for now we're just going to introduce that. So let's talk about how that 2,3-BPG is produced, how our body knows to produce it, and what conditions cause oxygen affinity versus decreased O2 affinity. So this unloading and loading of the O2 molecule is the respiratory movement, sometimes called hemoglobin dissociation. It is not directly proportional to the pressure oxygen or the partial pressure of O2 in the lung tissue, and it actually has a sigmoid relationship. Let's look at our oxygen or our oxyhemoglobin dissociation curve on the next slide, and we'll talk about some of the conditions that affect our respiration curve. All right, so this diagram is showing a representation of our oxyhemoglobin dissociation curve. Now let's first talk about what a normal oxygen dissociation is. In the lungs, when oxygen measurements reach 100 milligrams per mercury, the hemoglobin molecule is also nearly 100% saturated with oxygen. And in tissue where oxygen measures about 40 millimeters of mercury, the hemoglobin molecule is still about 75% saturated with oxygen. So that means that the remaining 25% was released to the tissue. And that's a normal metabolic or basal metabolic rate. And that is represented by the normal red line here. And I want you to think about it for a minute. If we only offload about 25% of the oxygen bound to our hemoglobin, this is one of the reasons why when we are resuscitating a person using CPR, we're still able to deliver oxygen through artificial respiration and through circulation with those chest compressions. So really important to remember, we don't offload all of our oxygen at once. We still have 75%, and so we can maintain oxygenation of a human being in the event of cardiac arrest if we have to, using those means, up to a point. Now, the artificial respiration is going to provide more oxygen into the lungs of that person, which will then allow prolongation of their life until we're able to stabilize them. So that's really important to remember. Okay, so we have this red line where our normal basal metabolic rate, but there are a couple of conditions that can cause a shift either one way or another. So let's take a look first at this green line, and we're going to call this green line a right shift. So some of the things that might occur or might be present during a right shift is we may have an increase of partial CO2, an increase of hydrogen ions present, increased temperature, and in this case we're going to have an increase of that 2,3-BPG, or in this case it's, remember, DPG. Those are interchangeable. We're going to have a more acidic pH, so a decreased pH. We may this might be present in a sulfhemoglobin situation, which we haven't talked about yet, but we're going to talk about when we talk about our abnormal hemoglobins. And this could also be present in hemoglobin, sickle cell anemia, or hemoglobin S is what we call it. And that's because of the abnormal shape and morphology of the hemoglobin S, or the sickle cell disease cells. They can't bind or offload appropriately. So what we're going to see is an increased need, but a decreased affinity, and so therefore an increased unload or offload of those oxygen molecules from the hemoglobin complex. I think it's really important for you to try to associate these conditions that I've mentioned with the right shift. And one of the ways that when I was a student, would remember that is I would always remember, gosh, it's hot in here, right? Increased temperature, right shift, and then you kind of, if you can remember one of them, you can kind of implement the memory or the memorization or the association with the other components that are present, or the other conditions that are present during a right shift of that oxyhemoglobin dissociation curve. When we look at a left shift, which is represented by this blue line on the left, we have sort of the opposite. All right, so we are going to have an increased affinity and therefore a decreased of oxygen offloading. We are going to have a decreased partial CO2 concentration, a decreased hydrogen ion concentration, a decreased temperature, as well as a decreased 2,3-DPG production. We are going to have an alkaline, or an increased pH in terms of our blood pH. Our PO is going to be decreased, and we might see this with methemoglobin, CO, or carboxyhemoglobin, and strangely enough, fetal hemoglobin. And this is actually a really great evolutionary advantage to for fetal hemoglobin, because if you think about the process of birth, it is traumatic both for mom and baby, and sometimes it can cause oxygen deprivation of a baby, but because they hold on to the oxygen, they hold on to it so long that it really only starts to be released when it is imperative, and therefore babies are able to transition better intrauterine to being born, so outside of the uterus. Words are hard today, you guys. I'm so sorry. All right, so moving on from that dissociation curve, I think it's really important for you to remember kind of the conditions. Now, let's think about some situations where a right shift may occur. You might have a right shift when you're running a race because you have an increased need of oxygen delivery to those muscles that are being used. When you travel to high elevation environments, they have a lower oxygen concentration in the environment there, or in the atmosphere there. And so actually, funny fact, when we do research or when we do testing on people who have grown and lived in high elevations, a lot of times they have a higher hemoglobin concentration naturally. Sometimes they have a larger lung capacity, and that is to compensate for the environment in which they've grown. All right. Interesting fact, in terms of the left shift, one of the criteria of resuscitation of drowning victims who have hypothermia, we cannot call a death on a drowning victim until we have brought their body up to a specific temperature. And that is because many times the metabolic needs of a person who is in a very cold condition slows so much that tissue demise occurs at a much slower rate. And a lot of times we can resuscitate people who have drowned in cold water and who have maintained a lower body temperature much more successfully than we can someone who has drowned in a higher temperature. So a lot of you've heard those, you might think that it's maybe an urban legend, but actually, it's actually true that a lot of people who drown in cold conditions and are hypothermic for long periods of time can survive underwater much longer than people who die in, the ocean where it's tropical weather. And this is why, because that dissociation curve reduces offloading, and that temperature has a lot to do with that. Okay, so we've done an overview of our dissociation curve, but let's talk about the right shift in a little bit more depth, because this is often one of the things we see more commonly in healthcare. Things like diabetic ketoacidosis, so we've got that decreased pH, so they're very acidic. And again, you guys are going to talk much more about this respiration curve or dissociation curve when you get into your blood gases unit with Jenny in chemistry. But let's talk about it. So in conditions of hypoxia, meaning decreased oxygen, we are going to see that shift to the right. The very first thing that's going to happen is we're going to have an increase in those 2,3-BPG levels, and that is going to result in hemoglobin having a decreased affinity for the oxygen. So we talked about affinity. So now they have a decreased affinity, meaning it doesn't like it. Affinity means to like or to have a need or a want for. Now all of a sudden hemoglobin doesn't want that oxygen anymore. It's going to kick it off with using that 2,3-BPG. Therefore, more oxygen is going to be released into the tissue. So under a normal basal metabolic curve, we release about 25%. But in this case, we're going to release a greater proportion of greater than 25%. And I do have this little YouTube video if you guys want kind of a visual on that. Optional, of course, but every little visual aid helps. On the other hand, when we have a left shift, that 2,3-BPG level is decreased in the red blood cell, and the hemoglobin molecules will hang on harder or more tightly to the oxygen. So we call that a tense form of deoxyhemoglobin. And then less oxygen is available for delivery to the tissues. So in this case, instead of 25% or greater than 25% of oxygen release, we are going to have less than 25%. And 25% seems to be kind of the magic number, okay? So we've got 25%, and then with a right shift, it's going to be greater than 25%, and then with the left shift is going to be less than 25%. So regulation of red blood cell production is going to be dependent upon a few things. Number one, conditions of hypoxia. So again, low oxygenation. If we have released all of the oxygen in the red blood cells and we still don't have enough, we become hypoxic. And that is going to stimulate the production of red blood cells within the bone marrow. And this is called erythropoiesis. So again, low levels of oxygen are detected by sensors in the kidneys. Interesting, right? So our kidneys actually play an imperative role of red blood cell production through the production of erythropoiesis, which is a hormone that signals the bone marrow to increase the production of red blood cells. So, yep, the kidney secretes that erythropoietin, it stimulates the bone marrow, and the bone marrow makes a priority of production of red blood cells because either we've lost it, through hemorrhage our red blood cell volume, or we are unable to oxygenate appropriately. Maybe we have a respiratory disorder, or maybe those red blood cells are being prematurely destroyed. Any one of those things could cause hypoxia, which will stimulate that erythropoietic process. I always like to tell my students that our body is amazing at compensating for difficulty. But sometimes there are things that can trick our body into thinking it's compensating, but it's really just being tricked. And in this case, we're going to see that with some of our abnormal hemoglobins of importance. Now, the reason why these are important is because they're common and they can cause health problems or sometimes demise in the case of severe events. So the first one is probably the most common, and you've probably heard about it. It's carboxyhemoglobin, and this is commonly referred to as carbon monoxide poisoning. This is where CO replaces the oxygen, and it binds 200 times tighter than the O2 to the heme and oxyhemoglobin. And so it's really hard to release that carbon monoxide from the red cells so that we can replace it again with oxygen. A lot of times people with carbon monoxide poisoning have to be put into a compression chamber to force that carbon monoxide to release, where oxygen can then bind to the heme again. The next is methemoglobin, and this is really more of an oxidative problem. Methemoglobin is iron that is oxidized to the ferric state. We really want our iron to be in the ferrous state, and that's because the ferrous iron is easily utilized, whereas ferric is more difficult to utilize in appropriate ways. This is usually caused by enzyme deficiencies, stress, exposure to environmental toxins. We do have a normal methemoglobin of less than 1% in circulation because there's always going to be some oxidative processes occurring. However, those enzyme deficiencies can cause a higher level of methemoglobin to be present in circulation, and we will talk more about those enzyme deficiencies as we move forward. The third type of abnormal hemoglobin is sulfhemoglobin, and that is where sulfur replaces oxygen. This could be due to ingestion of drugs, and unfortunately sulfhemoglobin is irreversible. It binds to the hemoglobin, and you cannot there's nothing you can really do to release that sulfur. It is just for the lifetime of that red cell bound. And so obviously if you have sulfur that has bound to your hemoglobin and it's circulating and not able to pick up oxygen, it's going to be problematic. So we've talked about all of the exciting things that make up a red blood cell and how they work and how they deliver oxygen, but how do red blood cells get energy? And why do they need energy? Well, I think it's kind of obvious. We've talked about the hemoglobin function. We've talked about the membrane integrity, deformability, ability for expansion with those fluid forces. But they also need to maintain a supply of ATP that enables them to facilitate those ion pumps on the surface membrane. Fun fact, red blood cells, again, are anaerobic because in their metabolism, they're anaerobic in their metabolism because they are anucleated and they do not have the mitochondrial activities that they once had when they were baby red blood cells residing in the bone marrow. So ATP is generated through a number of different pathways and of course anaerobically. The very first pathway that we see in red blood cell metabolism is the Embden-Meyerhof pathway. The second we know of is the hexose monophosphate shunt. Third is the methemoglobin reductase pathway, and then the very last one is the Lou-Behring-Rappaport shunt. Now, each one of these pathways has a different job. They are all metabolic pathways, but we're going to talk about each one and the specific role they play in maintaining the health and function of red blood cells. So if you think way back to your cell biology class, we're going to talk about glycolysis. Do you guys remember glycolysis from way back when? Well, the Embden-Meyerhof pathway utilizes glycolysis, and this is where about 90% of our ATP needed by red blood cells is made. So 1 mole of glucose makes 2 moles of ATP. It is non-oxidative, so it's anaerobic, and this is performed with mature red blood cells. So remember, they're non-nucleated or anucleated at this point. They've lost their nucleus. So don't be intimidated by this diagram. I love diagrams because I think that it really gives you a visual of how things are working. So here we start out with the Embden-Meyerhof pathway, utilizing glycolysis with glucose here. We get ADP, which turns into ADP. It's phosphorylized here, and then we have hexokinase. So I don't expect you to remember this diagram, but really what my point is here is I want you to see how one pathway moves into the next, okay? So the second pathway that I want to talk to you about is the hexose monophosphate shunt, okay? So that is where only about 5% of our ATP is produced in mature red cells. But the very important task that the hexose monophosphate shunt performs is it reduces NADP to NADPH. Okay? The NADPH is reduced glutathione or GSH, and it's the main defense for red blood cells against oxidative damage. Remember when we were talking about methemoglobin? Well, this hexose monophosphate shunt protects our red blood cells from that oxidative pressure, that oxidative process, keeping our iron that's within the hemoglobin in the ferrous state and not allowing oxidation to occur. If we have too little NADPH, intracellular oxidants accumulate and the globin in our hemoglobin can be denatured. We then see red blood cell inclusions called Heinz bodies, and we'll talk more about that as we get into kind of our red blood cell morphology and inclusions. But yeah, take a look at this diagram and just kind of see how everything fits together. All right? As you move forward and you're looking through your slides, if you need to come back and refer to this chart, please do. Okay, so methemoglobin reductase. So if oxidative damage does occur, methemoglobin reductase allows the body to get rid of that methemoglobin. A lack of methemoglobin reductase will increase methemoglobin levels, and there will be more ferric ions and less ferrous iron for that oxyhemoglobin. So let's talk about just some of the characteristics. We've got, so methemoglobin reductase maintains iron in the ferrous state to transport oxygen. If the environment is highly oxidative, there's constant oxidation happening within the body, and this kind of buffers it. Methemoglobin reductase, reduced oxygen carrying capacity if deficient, can be inherited or acquired by antibiotics. So there are a couple different things that can cause an incapacitation of our methemoglobin reductase. We could have an inherited tendency or pattern of inheritance from our parents. Some antibiotics can cause that, exposure to different medications or environmental exposures. It could be attributed to many different things, but the important thing to know is that this methemoglobin reductase keeps our iron in the ferrous state so that we can utilize it normally in our hemoglobin molecules for transport of iron. I mean, transport of oxygen. Okay, so the Lou-Behring-Rappaport shunt is our last line of, or last metabolic pathway. And this is really important because this is where our 2,3-BPG or DPG is produced. Now remember, without 2,3-BPG, our globin chains will not release the oxygen that is bound to our hemoglobin. It allows red blood cells to regulate that oxygen transport during conditions of hypoxia or acid-base imbalance or increased need. And then that 2,3-BPG in the red cell, oxygen is delivered to the body tissues with a decreased affinity of hemoglobins, of O2 to hemoglobin. So in that case, without 2,3-BPG that is produced through the Lou-Behring-Rappaport shunt, we aren't able to oxygenate appropriately. So each one of these metabolic pathways has a very specific job. We will go into a little bit more depth when we talk about enzyme deficiencies, etc. But for now, I really want you to know the name of each one of the metabolic pathways and their primary function. All right. So let's talk about a little bit about red blood cell senescence, and that just means red blood cell aging. It's a nice way of saying what happens when a red blood cell gets old. So a red blood cell travels about 200 to 300 miles in its 120-day lifespan. So it lives for about 3 to 4 months, usually around 4 months, but things can happen. And thus, each day we lose about 1/120th of our blood supply, so about 1%. And that's just the normal aging process of cells, okay? Each day we make about 1% of reticulocytes to recover. Now, reticulocytes, if you look at this picture here, they're going to be a little darker in color. And I always think about, if you think about a baby, they have kind of red, rosy cheeks. I always think of retics or reticulocytes as babies with red, rosy cheeks. Their color's a little bit more vibrant. They're a little bit rounder and fuller than the older, more mature red cells. And they don't always have a central pallor or this kind of cleared area of the biconcave disc shape. So they're kind of just fat and plump like babies, and that's because they are younger cells. They're sent out of the bone marrow at a younger age to compensate and to make up for the normal loss of red blood cells that we have. Now, we may see an increase of greater than 1% if we have a loss of blood and we have to compensate in greater numbers, and then we will have an increased reticulocyte count because again, our body is amazing at compensating for the need, and we see that pretty frequently. But we'll talk more about reticulocyte counts later on. As a red blood cell ages, we're going to see changes in the red blood cell indices. We haven't discussed the anatomy of our CBC with regard to red blood cell indices. That's going to be in our next PowerPoint, but take note of this and correlate it with the information that you get in the next PowerPoint. I will go over it in more depth. So some of the changes that we're going to see in the red blood cell indices of our CBC are, we're going to see an increase in MCHC as the red blood cell ages. We're going to see an increase in viscosity and density of our blood in general, or of the morphology in general, red blood cells. We are going to see agglutinability, and what that means is that sialic acid is going to start fading and degrading, and therefore it's going to start, our red cells are going to start losing their negative net charge, and they're going to start to stick together instead of repelling each other the way that they are meant to. We may see an increase in methemoglobin and sodium, an increase in oxygen affinity, which means it's not going to be able to release the oxygen in the way that it should to deliver oxygen to those tissues. With the loss of spectrin, which is, remember, the protein helps maintain the normal shape of the red cells, we may see a more spheroidal shape, so a spherocyte shape, which is going to be problematic when we are trying to go through circulation. It may be more susceptible to rupture and demise. And last but not least, we might see some membrane-bound IgG. So that means that our immune system has actually been able to identify abnormalities with the red cell because it is now aging, it's not functioning properly. There are, maybe some of the surface membranes that are immune regulating have disintegrated, and so now IgG don't recognize them as self, and they bind to the red cell membrane. We are going to see a decrease in enzymatic activity, a decrease in sialic acid, deformability, a decreased MCV, which is going to be mean cell volume or mean corpuscular volume, which is just referring to the cell's volume. We're going to see a decrease of phospholipid and cholesterol, which means that we are not going to have that same structure and shape that we need to for deformability and stability. We're going to see a decrease in potassium because those potassium pumps are not going to work appropriately due to decreased ATP. And last but not least, we are going to see a decrease in protoporphyrin, which means we are going to have abnormal heme development. So all of those things change as a red blood cell ages, and that can cause decreased function, which is why we need to have continuous reproduction or production of red cells in form of reticulocytes being output into circulation, so that we don't lose cells without replacing them. So what exactly happens when our red blood cells come to the end of their life? We have a very specialized system within the body that is highly acute and aware of the changes that are happening around them, and they pull out old and dysfunctional cells from circulation, and we call this the reticular endothelial system. Now, the reticular endothelial system is primarily comprised of splenic macrophages or resident macrophages of the spleen, but we do have some that occur in the bone marrow and also in the liver. So primarily spleen, but there are some of our reticular endothelial system in the liver and the bone marrow. And this is where normal extravascular hemolysis occurs. About 90% of red blood cell destruction occurs here, and these are aged red blood cells that are phagocytized by the reticular endothelial cells and digested by their enzymes. So what happens once we have broken down the red cells? Well, we recycle. Again, the body is very efficient in utilization of resources, and our transferrin, which remember is our carrier protein for iron, our transferrin returns that iron to the bone marrow, macrophages that recycle it and make more new hemoglobin in new cells. So let's talk about macrophages for a minute. Macrophages are white blood cells that reside in the tissues. They begin as monocytes, which we're going to talk about in our white cell unit, but they only spend a very short time in blood circulation, and then they go and reside in tissues, and they perform duties and jobs there. So in our reticular endothelial system, the macrophages that are resident there they pull aged and broken red blood cells out of circulation. The resident bone marrow macrophages actually serve as iron utilization, and we call them siderophages. After we break down the red blood cells, those globin chains are also returned to the amino acid pool. So the amino acids within those globin chain proteins are broken down and sent to be recycled. So we're going to reuse everything that we can to produce new red blood cells. So we've got our heme groups as well. Those are broken down. Of course, the iron is stored in the liver and the spleen as ferritin, and then the hemosiderin within those bone marrow macrophages. And then we do have a byproduct of biliverdin, which is then converted to bilirubin and excreted in the urine and the feces. So that's really the only thing that we waste, is that bilirubin. Everything else is recycled and reused for new cell production. Continuing on with the reticular endothelial system, so the protoporphyrin ring is going to be disassembled. Remember that protoporphyrin ring that is a part of the heme group. There is a carbon component associated with that portoporphyrin ring. It is going to be exhaled as CO2 waste. And then an opened tetrapyral ring, which we call biliverdin, is converted to bilirubin and carried by albumin, which is one of our other carrier proteins, to the liver for processing. Once that albumin has carried the biliverdin or the bilirubin to the liver, and the liver, the bilirubin is conjugated to bilirubin diglucuronide and excreted with bile into the intestines. And we're going to have a further breakdown once we get to the intestines. We're going to talk about what happens there in just a minute. All right, so once that bilirubin is conjugated and sent to the intestines, the normal flora or the bacterial action converts this to urobilinogen, or sometimes we call it UBG. Most of our urobilinogen is excreted in the stool, so that's what actually causes the color of our stool to be brown. But a small amount is reabsorbed through the enterohepatic circulation, filtered by the kidneys, and excreted into small amounts in the urine. So we are going to see a small normal amount of urobilinogen within the urine, and we measure that when we're doing urinalysis. We're going to talk about all of that when we get to our urinalysis unit, but just know that there is a normal small amount of urobilinogen present in the urine, and that is actually what gives it kind of that yellow color. Okay, so most of the red blood cell destruction that is a result of RBC senescence is done extravascularly through that reticuloendothelial process, but about 5% occurs within the lumen of the vessels, and we call this intravascular hemolysis, meaning destruction or rupture of the red blood cell within the lumen of our blood vessels. And that's really going to release the hemoglobin into the blood. So when the red blood cell is destroyed or hemolyzed, everything that is intracellular to that red blood cell is now going to be free floating in the plasma of our blood. So the hemoglobin is going to dissociate into alpha and beta dimers, and then that's going to be picked up by haptoglobin. Haptoglobin is another one of our carrier proteins, and it carries free hemoglobin, meaning hemoglobin that has been released from red blood cells during intravascular hemolysis, and is going to be transported to the liver for further catabolism. At this point, it's going to look very much like our extravascular process, but in this case, rather than albumin being the carrier protein, we are going to have haptoglobin. So in regarding intravascular hemolysis, there are a couple of points to remember. So the levels of haptoglobin are going to decrease in the plasma as it complexes with the free hemoglobin. So as free hemoglobin increases, haptoglobin levels decrease because they are all bound up with that free hemoglobin that's in circulation. We have 1 to 2 mils of red blood cell intravascular hemolysis, which can complete levels of haptoglobin. So that's not a lot. I mean, if you think about the minuscule amounts, but when you have, a good proportion of red cells that are being destroyed intravascularly, we can definitely see a disruption of our normal haptoglobin levels. When haptoglobin is completely bound up, free hemoglobin then appears in the urine. And any leftover hemoglobin is then oxidized to methemoglobin. So let's talk about this for just a minute. We are talking about intravascular hemolysis with regard to red blood cell senescence or aging, but there are other things that can cause intravascular hemolysis. We're going to talk about those in our hemolytic anemia unit, but just know that this isn't the only time when intravascular hemolysis can occur. There are many things that can cause destruction of red blood cells. In this case, it is at the age and maturity of the red blood cells, so the entire lifespan has been completed. But in the case of other hemolytic anemias, this is the destruction and disruption of red blood cell prematurely. So these, in this case, we're talking about aged red blood cells, but it can happen in other instances prematurely. As we come to the close of this first lecture on red blood cells and red blood cell metabolism, I definitely want you to do some further reading about protein carriers such as hemopexin. Hemopexin is just kind of a second line defense against free heme in circulation after hemolysis. It's kind of like second in line to protect heme-induced oxidative damage after haptoglobin. Read about whether hemoglobinuria, meaning hemoglobin in the urine, is always associated with hemoglobinemia, and which means free hemoglobin in the blood, and which substances, bilirubin or urobilinogen, appear in the urine and blood during which associated disease states. Now, if you don't get to all of that now, don't worry, we will cover that in more depth and more detail as we move forward. However, it's always good to kind of get a foot up just to kind of know what to prepare for. You guys are doing great. Thank you for joining me with our first lecture of hematology. And that is pretty much it for now. Bring back your question as needed. You have remained afloat. Way to go. I did also want to mention, go ahead and check out your objectives for red blood cell. Make sure you can answer all of the questions on your objective sheets that have been presented to you in your unit documents. And if you have any questions, please feel free to contact me via email, text message, or you can ask me in class the next day. I'm always welcome. I always welcome questions and conversations. Thanks again.